Hello to everyone. We will give a few minutes to wait for the rest of the participants, and we will begin soon. Thank you for your cooperation. Hello to everyone. We will begin our webinar. My name is Joao Batista. I'm a product manager in the area of microbiology at Conda Lab, and I'm going to be your host today in our webinar about cosmetic testing, focused on what microorganisms should we test in our cosmetic products. I hope you enjoy it and you learn a few things about cosmetic testing. Well, let's begin. What will we see today? Today we will cover a lot of ground, and we will start by answering the question: is microbiological control of cosmetic products necessary? To answer this question, we will offer you a brief market overview of the cosmetic sector, mainly focused on the Spanish market. We will touch upon the regulation and the safety evaluation of the cosmetic products, focused mainly in the microbiological quality and safety of the cosmetic products. And we will talk about the standardization that is experimenting this sector in the last few years. After that, we will see the risk associated with to the cosmetic products. We'll offer you a brief definition of the term cosmetic product. We will see that it's not what you have in mind. Then we will talk about the microbiological risk assessment. We will touch upon the factors involved in the risk assessment of the cosmetic products. And we will talk about briefly of the preservative added to those cosmetic products that need them. After that, we will talk about the microorganisms that are tested in the cosmetic products, and we will expand on the mesophiles, molds, and yeasts. We will talk about the specific microorganisms that are collected in the ISO standards of microbiology of cosmetic products, and we will talk about other microorganisms of interest in the industry that are not in the ISO standards, but are increasingly important in the cosmetic sector. After we've seen all the methods of microbiological analysis, we will offer you the best alternative in the market by the hand of Chrome line of products. I hope you enjoy it, and we will begin with the presentation. First of all, we will talk about the question, is microbiological control of cosmetic products necessary? Well, to answer this, we will have to start by taking a look of the cosmetic sector, mainly in Spain. The cosmetic sector in Spain generated 8.2 billion euros last year, and it experimented a growth of almost 8% in the last year, as I said. Only the perfume sector, a subsector of the cosmetic sector in Spain, generated 5.3 billion euros last year, and it places Spain as the #2 exporter of perfume and cosmetics in the world. The cosmetic sector also has a great importance in generating jobs in our country. The sector produces more than 40,000 jobs directly and more than 250,000 jobs indirectly. In recent years, this sector, the cosmetic sector, has experimented a huge growth due mainly to the greater interest in personal care of the population. Everybody in our country and similar countries uses between 7 and 9 cosmetic products a day and spends almost 170 euros a year only in cosmetic products. This huge importance in the economy and in the society of our country and many others has promoted a trend towards regularization in the sector. This regularization is materialized in regulations such as the regulations number 1223 of 2009 on cosmetic products by the European Council. It gives a regulatory framework that manufacturers and other parts involved in the cosmetic industry can adhere to standardize their processes. In Article number 2, this regulation provides a detailed description of what a cosmetic is, and we will touch upon that later. It also regulates the role of all the parts involved in the production of cosmetic products, such as the manufacturer, importer, distributor, and even the customers. This regulation also recollects the obligation of making a file that contains the product safety information and a file that addresses the microbiological specifications of the cosmetic product. This file has to be preserved during 10 years after the commercialization of the cosmetic product. The appearance, the resurgence of these regulations also comes with a trend toward standardization. That stems from the need for harmonization in the production and manufacturing processes and the quality and safety control in the cosmetic sector. This standardization is present in every step of the production, design, commercialization, and many other steps of the cosmetic development. The emergence of ISO standards standardize the risk assessment and microbiological testing of the cosmetic products, and it provides a series of benefits, such as increased safety and reliability in the manufacturing and safety and quality control processes. It gives support for public policies and legislation. It increases the interoperability between companies of the same sector, and it facilitates the market access to new companies. It fosters innovation and gives increasing awareness of new initiatives and technical advances in the sector. After this, we will talk about risk associated to cosmetic products. But first, we need to know what is a cosmetic product. The European regulation number 1223 2009 that we talked before says that a cosmetic product is any substance or mixture intended to be placed in contact with the external parts of the human body, such as the skin, the teeth, the mucous membranes, etc., with the main purpose of cleaning them, perfuming them, and changing their appearance, also protecting them, etc. So, when we think about a cosmetic product, we mainly think about moisturizing cream, makeup, etc. But the term cosmetic product encapsulates a huge array of different products, such as oral hygiene products, hair care products, perfumes, soaps, etc. So the term cosmetic product encapsulates a lot of products with different characteristics that have to be taken into account when performing the risk assessment and the microbiological analysis. The risk assessment is performed following the ISO 29621 of 2011, and its main purpose is identifying those cosmetic products of low microbiological risk. But first, what is microbiological risk? It is the ability of a product, of a cosmetic product, to be precise, to support the growth of microorganisms and the likelihood that those microorganisms may cause harm to the user, and also the ability to support the presence of those specific microorganisms recollected in the ISO standards of cosmetic microbiology. So, by this ISO standard, the risk assessment is based on the following factors. First of all, the product composition. This is important because every product has its own characteristics, and some factors such as the pH value and the activity of water of the cosmetic product might interfere with the potential growth of microorganisms in the product. Also, the use of raw materials that can create a hostile environment can interfere with the growth of microorganisms. Raw materials such as ethanol may interfere with the growth of microorganisms by killing them or by slowing their growth. The production conditions also play a huge part in the risk of a cosmetic product, because if to manufacture a certain product you need high temperatures during a long period of time, you can kill or hinder the growth and development of the microorganisms that might be present in the product. Next, the packaging is also important because some types of packaging might slow or even prevent the contamination of the cosmetic product. Monodose packages and disposing pumps in the cosmetic products may stop the growth or the contamination of the product. And finally, the combination of any of these products have a synergic effect and can increase or decrease the risk of contamination of a certain cosmetic product. Based on these factors, we can distinguish two types of cosmetic products: those that present a low microbiological risk and those cosmetic products that have a high microbiological risk. What are those products of high microbiological risk? These are the products that are highly susceptible to microbiological contamination and that require exhaustive microbiological control and the addition of preservatives. These high-risk microbiological products are regulated mainly by two ISO standards: the ISO 21148 that recollects the general instructions for microbiological examination, and the ISO standard 17516 that has written the microbiological limits of certain microorganisms in our cosmetic products that we will touch upon later. Before talking about the microbiological analysis, we will have to stop a little bit in the preservatives. These are substances that are added to the cosmetic product with the main purpose of inhibiting the growth of microorganisms that may be present in our product. These preservatives are incorporated into the formulation of the cosmetic in very small concentrations during the manufacturing processes. The function of these preservatives is to mainly prevent the microbiological contamination of the product, but they are never intended to destroy the microorganisms that can be present. So they only slow the growth or stop the growth of microorganisms, but never destroy them, because these can hide problems during the manufacturing processes that need to be addressed. So after all this, we will talk about what microorganisms are tested. To ensure the quality and safety of the cosmetic products, the microbiological control is standardized by the ISO standard 21148 and is performed according to a risk analysis, taking into account the factors that we talked before. Prior to the detection or counting of viable microorganisms in a cosmetic product, we will have to first neutralize the preservative that we have added to the formulation to stop the inhibition of potential microbial growth. If we don't do this, if we don't neutralize the preservative added to the cosmetic product, we will not see the true results or the true growth of microorganisms present in the sample. So first of all, we have to neutralize the preservative added to the cosmetic product. Well, cosmetic products are not considered sterile, so they can contain a certain number of microorganisms, but this number of microorganisms should not be excessive. The main microorganisms tested in cosmetics are viable microorganisms, such as aerobes and mesophiles, molds and yeast, and specific microorganisms, such as pathogens like Staphylococcus aureus, Pseudomonas aeruginosa, and Candida albicans, indicators of contamination and bad hygienic practices during the manufacturing process, such as E. coli, and other microorganisms that are not recollected in the ISO standards, but are of increasing importance in the industry, such as Burkholderia cepacia, bacteria of the genera Clostridium, other molds, and other enterobacteria. The limits of detection are recollected in the ISO 17516 and are the next. For products intended for use for mainly children under 3 years of age to be applied around the eye area or mucous membranes, the limit of detection of mesophile aerobic microorganisms such as bacteria, yeast, and molds are under 100 colony-forming units per gram or milliliter of cosmetic product. For other products, the rest of them, the limit of detection is under 1,000 colony-forming units per gram or milliliter of product. And for pathogen or indicator microorganisms, we need to have an absence of any microorganisms in 1 gram or milliliter of product. Well, now we will see the methods of detection or enumeration of the microorganisms that we talked before. First of all, we will talk about viable aerobic and mesophilic microorganisms, which are performed following the ISO standard 21149. The main focus of this analysis is to detect aerobic microorganisms. Those are those microorganisms that can grow in the presence of oxygen, and those microorganisms, the mesophilic microorganisms, that can grow in a range of temperature between 20 degrees Celsius and 45 degrees Celsius. The analysis is the following. We take 1 gram of our sample or cosmetic product, and we will perform a dilution, a 10-fold dilution in a neutralizing medium, such as the English broth, to neutralize the effect of the preservative added to the cosmetic product. Then we will wait for 20 minutes at room temperature and inoculate an agar plate of tryptic soy agar, or more commonly known as TSA. And then we incubate the plate at 32.5 degrees Celsius during a period of time of between 48 and 72 hours. And then we perform the colony count, which gives us an idea of the total microbial load of our cosmetic product. Next, we perform the detection and counting of molds and yeasts following the ISO standard 16212. And our main focus here is to detect molds that are multicellular fungi and yeast, which are single-celled fungi. These fungi produce spores that are mainly resistant to desiccation, but not to heat. And these microorganisms can also ferment the cosmetic product, and they also can change their appearance and their properties. So it's very important to analyze these microorganisms. The analysis is very similar to the last one. We take a gram or a milliliter of our cosmetic product. We perform the neutralization in our neutralizing broth, such as the English broth. We wait for 20 minutes at room temperature, and we inoculate a plate of Sabrot dextrose chloramphenicol agar, which is special for molds and yeasts. And we incubate the plate at a temperature of 25.5 approximately degrees Celsius during a period of 3 or 5 days to permit the growth of these microorganisms that are quite slow. Then after the incubation, we perform the colony count, and we have an estimation of the molds and yeast load in our cosmetic product. Next, we will analyze our specific pathogens. We will begin by analyzing Staphylococcus aureus. This bacteria is a gram-positive cocciform bacterium that forms three-dimensional structures similar to a cluster of grapes, which gives them their name in Greek. It is an ubiquitous bacterium that is present in epithelial tissue of individuals, mainly the mucous olfactory membranes of humans and the epidermis, mainly the skin of healthy adults. The infections of Staphylococcus aureus can occur by direct contact with a contaminated product or object, or by inhalation of droplets and aerosols containing the bacteria. Staphylococcus aureus can produce skin infections that may cause blisters, abscesses, redness in the area, and swelling. The analysis is performed following the ISO standard 22718, and as the last analysis, we take 1 gram or milliliter of our cosmetic product. We neutralize the preservative in our neutralizing medium, such as the English broth. And now we incubate this initial suspension at 32.5 degrees Celsius approximately between a minimum of 20 hours. Then we inoculate a plate of Bayer Parker agar that is supplemented with an emulsion of egg yolk and potassium telluride. Then we incubate the plate at 32.5 degrees Celsius approximately during a minimum period of 24 hours. And then we identify the suspicious colonies. In the case of Staphylococcus aureus, the colonies are of black color due mainly to the reduction of telluride, and they have a clear halo due to the activity of the lipase and lecitinase enzymes of the bacteria. Here we have them in a real plate, so you can see them. Those are the colonies of Staphylococcus aureus. Next, we analyze the next bacterium, Pseudomonas aeruginosa. This one is a strict aerobic gram-negative bacteria shaped as a bacillus. This bacteria is present in soil and water all over the planet, and they grow in humid areas, such as medical instruments and humid facilities. They can also grow in expired or inactivated antiseptic solutions, so its analysis is very important. Pseudomonas aeruginosa can produce infections that range from mild external infections, such as ear canal infections, and they can also produce severe life-threatening illnesses. So the analysis of Pseudomonas aeruginosa is very important in our cosmetic products. The analysis is performed following the ISO standard 22717, and as the last one, we take 1 gram or milliliter of our cosmetic product. We inoculate it in the same neutralizing medium, the English broth. It's the same dilution as the last one, a 10-fold dilution of the product. And then we incubate the initial suspension, same initial suspension, at a minimum of 20 hours at 32.5 degrees Celsius. Then we inoculate a plate of Cetrimide agar, and we incubate this plate at 32.5 degrees Celsius again, now during a period of 24 hours minimum. Then we identify the suspicious colonies. In this case, in the case of Cetrimide agar, the colonies of Pseudomonas aeruginosa are bright yellow color, due mainly to the production of a pigment called fluorescein that has a bright yellow color during under natural light, but is fluorescent under fluorescent light. Here we can see a plate of Cetrimide agar, and we can see the bright yellow color and the bright yellow halo that produces Pseudomonas aeruginosa. Then we confirm the suspicious colonies by inoculating a plate of King A agar, specific for Pseudomonas aeruginosa, and we incubate this plate again at 32.5 degrees Celsius during another 24 hours. In our confirmation medium, our King A medium, we can see that the colonies of Pseudomonas aeruginosa are mainly greenish yellow color, but now they have tinted the medium with a blue pigment that is very characteristic of Pseudomonas aeruginosa. You can see it in this plate. Well, next in line, we have Candida albicans. This is a dimorphic fungus. It develops in two different ways depending on the temperature. It develops as a yeast at the body temperature of the host, mainly 37 degrees Celsius, and at room temperature, such as the temperature found in nature, like 25 degrees Celsius, it develops as a filamentous fungus. It is a ubiquitous yeast that resides mainly in the skin of healthy individuals, mainly harmlessly, but when the immune system of the host is altered and the conditions such as humidity and heat permit its growth, it causes infections. The infections caused by Candida albicans are called candidiasis and are infections of the skin and mucous membranes. They are mostly found in the skin folds, interdigital spaces, genitals, cuticles, and oral mucosa, and it provokes swelling, redness, and when it affects the mucus of the mouth, it has a very distinctive white secretions. The analysis of Candida albicans in cosmetic products is performed following the ISO standard 18416. And as the last one, it begins by taking 1 gram per milliliter of our cosmetic product. We perform the same dilution in the same neutralizing medium. It's the same initial suspension as the last analysis. It's incubated at 32.5 degrees Celsius during a minimum of 20 hours. And then we inoculate a plate of sabouraud chloramphenicol dextrose agar, specific for yeast and other fungi. And we incubate this plate at 32.5 degrees Celsius during a minimum of 24 hours. After the incubation, we can identify the suspicious colonies in our sabouraud dextrose agar with chloramphenicol. That, in the case of Candida albicans, are white colonies. And then we perform the confirmation of these suspicious colonies. We do this in two different ways. First, we perform a germination test that's mainly focused in detecting the appearance of germination tubes by Candida albicans. And this is performed by inoculating a tube of fetal bovine serum with a suspicious colony, incubating it at 25 degrees Celsius or room temperature or 35 degrees Celsius, sorry, during a period of 2 hours, and then inspecting the growth of germination tubes under the microscope. Then we take another suspicious colony and we inoculate a plate of cornmeal agar to confirm these suspicious colonies. We incubate this plate for another 24 hours at a temperature of 32.5 degrees Celsius approximately, and we can identify then the colonies of Candida albicans under the microscope by the production of chlamydospores that are very refringent structures that can be seen clearly under the microscope. Next, we have a specific microorganism. This time it's not a pathogen, but they can be. E. coli, or Escherichia coli, is a facultative anaerobic gram-negative bacillus and is a commensal in the gastrointestinal tract of humans or homeothermic animals. Most E. coli do not cause problems. They are present in our microbiome and they don't cause any harm. But some types of E. coli can produce some toxins, like the toxins that the bacterium of the genera Shigella produces, called Shiga toxins, and they can cause sickness, diarrhea, and many other symptoms. Due to their high presence in the intestinal tract and feces of humans and animals, E. coli is an excellent indication of poor hygienic practices during the manufacturing of the cosmetic products. So the analysis of E. coli is performed following the ISO standard 21150 and is done the following way. First of all, we take a gram or a milliliter of our cosmetic product, and we perform the same dilution in the same neutralizing medium as we've talked before. We incubate this initial suspension at 32.5 degrees Celsius during a minimum of 20 hours, like we did before in the other analysis. And then we inoculate a plate of McConkey agar. Then we incubate the plate for 24 hours at a temperature of 32.5 degrees Celsius approximately. And then we identify the suspicious colonies. In the case of E. coli in McConkey agar, the colonies will be a bright red color, and they will have a very characteristic halo around them. The halo is produced by the drop of the pH values around the colonies of E. coli, mainly due to the fermentation of lactose in the medium. Here in the picture, we can see the aspect of the colonies of E. coli in the McConkey agar plate. Then, to confirm the suspicious colonies, we inoculate a confirmation medium, such as Levine agar, and then we incubate the plate at a temperature of 32.5 degrees Celsius, like before, during a minimum period of 24 hours. After this, we can confirm the colonies of E. coli in Levine agar by the appearance of the colonies. That is, they are dark colonies, mainly green, slightly bluish greenish tint with a metallic green appearance. We can see that in the picture. We can see the metallic green colonies of E. coli. Next, we will see an emerging pathogen that is not recollected in the ISO standards, but its increasing importance puts in perspective the need of analysis under the laboratory. Burkholderia cepacia is a gram-negative aerobic bacteria present in soil, water, and some plants, and it is considered an emerging pathogen in the recent years, mainly due to these factors. Burkholderia cepacia can survive in many different habitats and under very diverse environmental conditions. It can also resist some antimicrobial compounds. It also causes opportunistic infection in people with cystic fibrosis and some immunodeficiencies. And the analysis of Burkholderia cepacia is performed under the United States Pharmacopoeia. Next, we have the bacteria of the genera Clostridium, mainly Clostridium botulinum and Clostridium perfringens. These are gram-positive, obligatory anaerobic bacteria that can produce spores that are heat resistant. In the case of Clostridium botulinum, they can produce a very dangerous toxin, and they can grow in airtight containers. In the case of Clostridium perfringens, it inhabits the gastrointestinal tract of humans and animals and can be found in the perianal skin of healthy individuals. So they can be used as an indicator of bad hygienic practices during the manufacturing process of cosmetic products. The detection of these bacteria is performed using the recommended method of the FDA, the Food and Drug Administration of the United States. Next, we have other filamentous fungi, such as the Penicillium and Aspergillus fungi. They can produce allergies and secondary mycosis, and in the case of Penicillium, it's one of the most predominant genera of filamentous fungi in cosmetics or for skin application, so their analysis is also very important. And Aspergillus, that are frequent in all types of products, but the most important species of this genera is Aspergillus flavus and Aspergillus parasiticus that can produce aflatoxins. Aflatoxins and other mycotoxins that are produced by filamentous fungi have many adverse effects in the organisms of humans, such as neurotoxic, nephrotoxic, hepatotoxic, and they are potentially carcinogenic compounds. Next, we have other enterobacteria that can serve as indicators of fecal or bad hygienic contaminations in cosmetic products. Apart from that, they can ferment the compounds of our cosmetic products, and they can alter the composition and the aspect of the product. So their analysis is also very important. Here we have a list of them, such as Serratia marcescens, Pluralibacter gregoviae, the bacteria of the genera Klebsiella, Enterobacter, Enterococcus, Proteus, etc. Well, now we have seen the standard methods of detection of microorganisms in cosmetic samples, and we will present you our Condochrome line of products that offers you the best alternative to this analysis. What is Condochrome and why should you use it? Well, first of all, our line Condochrome of products gives you faster results than the traditional methods. In only 24 hours after inoculating your plate, you will be able to see your results. These results are of easy interpretation because you can identify your microorganisms by their characteristic color. Just by giving a simple glance over a plate, you can see which microorganisms you have. The Condochrome line of products also saves you a lot of time and space because you can detect many different microorganisms in just one plate. And also you can perform almost all the analysis you need simply in one plate. Our line of products also has an increased selectivity and sensitivity. You can detect very specifically the bacteria you're looking for, and you will be able to detect these bacteria with a very low concentration. So what happened? Sorry. Wait a minute. Sorry. Our line of products also requires a minimal investment because you don't need to purchase any additional equipment for your lab or train your personnel because the interpretation of results is very intuitive. The first product in our line Condochrome that we will present you today is our product Condochrome PEG. With Condochrome PEG, you can detect 3 of the 4 main microorganisms of obligatory detection in cosmetic products, such as Pseudomonas aeruginosa, Escherichia coli, and Candida albicans. The detection, the simultaneous detection of these 3 microorganisms will save you a lot of time and resources in your lab. This method has been validated by INEA following the requirements of the ISO standard 16140, and the method is the following. Using the same initial suspension in neutralizing medium that you have made for the rest of the analysis, you can take you can inoculate a plate of Condochrome PEG agar, and by incubating it only 24 hours, you can see your results. You can identify the 3 microorganisms simultaneously in only one plate. The E. coli bacterium appears as red colonies. Candida albicans can be seen as blue colonies, and Pseudomonas aeruginosa produces yellow beige colonies. So the detection is very, very intuitive and very simple. And only in 24 hours, you can detect these 3 microorganisms. The next microorganism of obligatory detection by the ISO standards is Staphylococcus aureus, which you can detect using our product Condochrome Staphylococcus. It allows you to detect the Staphylococcus bacterium in only 24 hours. Our product also allows you to detect many other species of Staphylococcus in your sample if you need to. The method is the same as before, using the same neutralizing medium, pre-incubated. You inoculate a plate of Condochrome Staphylococcus agar, and you incubate it at 32.5 degrees Celsius during a minimum of 24 hours, and you can identify the colonies of Staphylococcus aureus with a bright red yellow color. So in only 24 hours and using only 2 agar plates, you can identify the 4 obligatory microorganisms recollected in the ISO standards. So you can simplify the process of detection in your laboratory by using our Condochrome line of products. But if you want to go a step further, you can also detect the emerging pathogen Burkholderia cepacia using our Condochrome Burkholderia product that detects Burkholderia cepacia in cosmetic products. This method has also been validated by ENIA as an alternative to the USP method. The process is the same as before. Using your neutralizing medium, pre-incubated, you inoculate a plate of Condochrome Burkholderia medium, you incubate it for only 24 hours at 32.5 degrees Celsius, and you can detect the colonies of Burkholderia by their characteristic color, that is a very intense brown color. You can detect it very simply by looking at the plate. So by only using 3 agar plates, you can detect all the microorganisms that you need in your cosmetic samples. Well, we have reached the end of the presentation. I'd like to thank you for your attention, and now we will begin our questions and answers or Q&A. So feel free to ask any questions you need, but if you have any doubts or you need any information about our Condochrome line of products, feel free to message us at our email address, micro@condalab.com. I repeat, you can send us any questions you need at our email address, micro, M-I-C-R-O, @condalab.com. Now we will begin our Q&A, so feel free to ask anything you want. Well, don't be shy. We will answer any questions you need. Let's see. Well, I hope you learned a lot today. Well, we have our first questions. Let's see. Here they ask, can we use the same initial suspensions for all our analysis? Yes, we can use the same initial suspension for analysis with our neutralizing broth. We perform a 10-fold dilution of our cosmetic product, and then we can inoculate all the mediums we need. For example, if you want to use our Condochrome line of products, you perform the 10-fold dilution in your neutralizing medium, such as the Ingley, and then you can inoculate the plate of Condochrome PEC, the plate of Condochrome Staphylococcus, and then you can inoculate the plate of Condochrome Burkholderia if you want to. But if you want to perform any other analysis, you can use the same initial suspension. Well, let's see. Other questions. They say, which dilution do we need to perform the moles and yeast count and the aero and mesophiles count? There is no pre-established dilution that you can perform. Mainly due to empirical analysis, you can perform a 3-fold or 5-fold dilution, but if you need to, you can dilute it further up to whatever dilution you need. You have to take into consideration that for a plate to be countable, it needs to have less than 300 colonies. So if you perform a 5-fold dilution and you see that the plate is still uncountable, you can perform further dilutions if you need to. Let's see. Other questions? Okay. They ask us which samples were used during the validation of Condochrome PEC. For the validation of Condochrome PEC, we used a vast amount of samples, mainly to cover the many different kinds of cosmetic products that are in the market. As we said before, the term cosmetic product encapsulates a lot of different products. So to take into account this huge variety of products, we use products in 5 categories mainly. Cleaning and hygiene products such as toothpaste and hair care products, etc. We use gel products such as gel soap, hand soap, gel shower, shampoo, etc. And we use sunscreen, also another category of wash of sunscreen, because sunscreen is also a cosmetic product. We use makeup because makeup is the main cosmetic product that comes in mind when you think about a cosmetic product. And we use emulsions such as a moisturizing cream, etc. So we covered a lot of ground when validating the Condochrome PEG line of products because the intended use of this product is with any cosmetic that can be available in the market. Very nice question, by the way. Then they asked us, let's see, we have many questions. They are asking us if there is no standardized method of detection for Burkholderia right now. At the moment, there is no standardized method of detection for Burkholderia. It is performed following the recommendations of the USP, the United States Pharmacopoeia, but at the moment, there are no standards for the detection of Burkholderia. For example, the product Condochrome Burkholderia has been validated by INEA by checking the detection process performed in many different laboratories and establishing an average or a standard method of detection. But at the moment, there is no official method of detection. So the Condochrome Burkholderia product can be the best alternative in the market to analyze Burkholderia cells. Well, I see that we have a lot of questions, but sadly we are running out of time. So if you want to ask any further questions or just to answer the questions that have been pending, you can feel free to ask to write to us at the email address micro @condalab.com, and we will be more than happy to answer any of your questions. Thank you so much for your participation and for your attendance to our webinar, and we hope to see you in the next one. Have a nice day, and thank you so much.